Okay, so I wish I knew how to turn off the beeps, but I'll figure that out for next time. This is our first ever seminar in the biological stoichiometry seminar series. And I'd like to point out this is a production of Ratios Matter, which is a stoichiometry newsletter. If you're interested in that, then send me a message and I can direct you to our website. So just a little bit of background on the stoichiometry seminar. We've seen a number of online conferences and seminars sort of emerge over the last year during the pandemic. And, obviously this is a great opportunity to share science more widely outside of the university or institute or company that you're at. It's a chance for us to talk about emerging issues and give younger scientists a chance to showcase their work and to build a more robust network of colleagues around the world. So we would like to have this seminar do sort of like all of this. We hope this is going to be a gathering place to discuss stoichiometry with like minds. Our current plan is to host about one seminar a month and keep this going if there's sufficient interest. We have seminars already scheduled from now until the middle of next winter, so it seems like we have at least can find people to give the talks. Hopefully people will show up. The talks will continue to be advertised through Twitter, and we can send you an email with a Zoom link if you want, so that you can receive notifications of upcoming talks. And we're also live streaming this. Hopefully that is working. That live stream is recorded and it will be available in the future. So if you want to see this again or share it with someone else, there's a way to do that as well. We see the seminar as being flexible. So it could be a 45-minute presentation, kind of like a departmental seminar, or maybe it's a collection of shorter conference-style talks, multiple talks over the course of an hour, or maybe something conversational with like a panel, or maybe it's a shorter talk with a longer discussion period where we really sort of hash out some new results. So we're really open to these ideas and suggestions. If you have something that you think might work or want to talk to us, just drop us a line. And finally, before we get to the speaker, I'd just want to thank all of my co-creators at Ratios Matter for helping make this happen. I'm not a one-person show, and this is only possible with contributions from the whole team. And in particular for this seminar series, we have a seminar series group, which includes Jess Corman, Nicole Wagner, and Kat Jones, and they've all been instrumental in making this happen. So with that, I'm very pleased to introduce our first speaker in the seminar series, Kimberley Lemmen. Kim is joining us today from Switzerland, where she's recently started as a postdoc at the University of Zurich. And before that, she completed a master's at Queen's University in Canada. And she's nearly finished with her PhD, which she completed at Netherlands Institute of Ecology. Her defense date has been set for June 14th. So let's just all remember to send good thoughts towards her way in about a month. Kim is generally interested in using microorganisms to understand the predictability of organismal responses to environmental change, including changing supplies of elements. She has a bit of a stoichiometry background, as she was a group leader at Woodstock 4 conference that was held in Montana. That was pre-pandemic days, back in the good old days of 2019. And today she's going to be talking about the evolutionary responses of primary consumers and how this reduces the cost of stoichiometric imbalances. And with that, I'd like to welcome Kim and thank you for being our first speaker.
Okay, thanks, Paul, for that really nice introduction. So yeah, today I'll be presenting work that I did during my PhD at the Netherlands Institute of Ecology. So yeah, I'm based out of Zurich now, but all the work that I presenting today was done there. And I really want to thank everyone at NEO up top for all the support that they provided me throughout my PhD. So when I was asked to participate in the seminar series, I thought it was this amazing opportunity because actually the first conference that I attended during my PhD was the conference on biological stoichiometry, which was held at Paul's home university in Trent. And throughout my PhD, I've received such fantastic support from the stoichiometric community, both my peers as well as mentors within the field. And I couldn't imagine a better way to be finishing off my PhD. So I just really wanted to also thank the organizers of this seminar series for inviting me to participate. Okay, so most of us are familiar with the concepts of stoichiometry, but for those who are not, I'll give a very upfront high-level overview. So biological stoichiometry provides a method to study biotic and abiotic interactions by breaking these interactions down into their elemental building blocks, and then using the stoichiometry, so that is the relative abundance of elements to one another, both within organisms as well as the environment, to understand patterns of biodiversity. And so this framework has been really successful in advancing understanding in ecological patterns and processes. And so because of that, it's been suggested and that it help understand evolutionary patterns and trajectories. And these are things that have been like pinpointing selective agents is something that's really notoriously difficult to do. And by breaking these down into elemental building blocks, that might be successful in understanding these patterns. And so one thing that I'm interested in is stoichiometric mismatches. So, and how they may act as a selection pressure. So a stoichiometric mismatch occurs when an organism is not receiving what it needs to grow, survive, and reproduce from its environment. So one example occurs between autotrophs and primary consumers. So autotrophs have quite flexible body stoichiometry for a variety of physiological reasons, and they often reflect their elemental composition often reflects what's available in the environment. So for example, here you've seen this green algae, which the ratio of carbon to phosphorus can range anywhere between 100 to 2000. And so these organisms can have are very flexible, but in contrast, the next trophic level up with the primary consumers have far more constrained body stoichiometry, and they require a very specific set of elements and relative abundances in order to be successful in their environment. And what can happen is that if the food that primary consumers are eating deviates too much from what is optimal in their environment, they can have decreased fitness. So you can see whether or not these ratios are too high or too low that both depress organismal fitness. And so the study by Zhao and de Klerk shows that both population growth rate and somatic growth rate are affected by stoichiometric mismatch. And so this had led me to be interested in whether or not these stoichiometric mismatches can be a driver of microevolutionary change within populations. And the reason why microevolutionary changes may be occurring is also because we're dramatically altering the availability of elements globally. So pictured here, we see a phosphorus mine. And so what's happening here is that phosphorus usually becomes available through natural processes such as weathering. But this mining liberates large amounts of phosphorus, which can then be inputted into the environment, leading to things such as eutrophication. And so not only are humans changing how much elements are going into the environment and their availability, but in response to seeing the negative impacts of increased nutrient availability, municipalities and municipalities have put in legislation to decrease the amount of nutrients that can be available, which has led to re-oligotrophication in many systems, restoring it to more natural states. But this also means that nutrient availability has changed a lot within a relatively short period of time. And so these dramatic changes in biogeochemical cycles may lead to even greater stoichiometric mismatches within systems. And so I'm interested in how organisms respond to such stoichiometric mismatches. And so the study system that I've used to tackle these questions is the experimental evolution. And so with these studies, you look at evolutionary changes that occur within experimental populations as a consequences of conditions imposed by the experimenter. So for example, here we can see that we have this ancestral phenotype, and after the experimental evolution, that these phenotypes change over time in response to those selection pressures. And so for my work, what I do is I use the microzooplankton, Brachionis caliciflorus. And so this is a microzooplankton that's part of the rotifer phylum. And so the reason why it's great for these experimental evolution studies is that it's relatively easy to culture. So we can have very large population sizes, which is great for anything related to evolution. And we can easily manipulate them. So we can move them around, we can change their culturing regime, we can apply different selection pressures. So they're really useful for these studies. Additionally, they have very short generation times, and that allows us to observe evolution more quickly than say with other organisms such as Daphnia that have generation times of a couple of weeks. These are just a couple of days. And finally, these have a unique life cycle. They're cyclical parthenogenics. And so this means that they reproduce both sexually and asexually. And this has advantage for experimental evolution studies because sexual reproduction allows for recombination. So you can have the mixing of different like genetic components, which can allow for adaptation to occur. And additionally, we have this asexual phase of reproduction where population sizes can grow quite quickly. And this allows us, again, to have these large population sizes. And so in this experimental system, what we do is that we take our focal study species, this Brachionis caliciflorus, and then we apply different selection pressures. So these selection pressures can be anything from the culturing regime, which allows us to select for fast population growth or competitive ability. Then we can apply abiotic stressors, as well as biotic stressors such as a stoichiometric mismatch. And so all of these stressors can be applied either in isolation or in combination with one another. And so because I was interested in how organisms respond to stoichiometric mismatches, the first thing that I needed to do was determine whether or not there was a potential for microevolution in stoichiometric traits. And so the way that I thought would be best to go about doing this was to quantify the amount of intraspecific variation in such stoichiometric traits. And the reason for this is because standing genetic variation is considered the fuel for rapid adaptation. For example, if we have a genetically diverse population here, each different color represents a different genotype. And if we have a dramatic environmental change within the system, the more diverse your population is, the more colors that you see here, the greater the chance that one of these genotypes will perform well in this new environment. And so what we can see is that we have this green genotype take off, and we will see a shift in the mean phenotype of the population that has a genetic component, and thus we see a rapid evolutionary change within this system. So to investigate this question, I did so at the Woodstoik 4 workshop. So this was led by Jim Elser here and Michelle Evans-White. And so this is a workshop for early career researchers, specifically those interested in ecological stoichiometry. And this is a workshop that basically allows young scientists to come together, generate an idea that we think is important for advancing the field of ecological stoichiometry, and generate a manuscript based on this idea in a fairly abbreviated time frame. And so in this Woodstoik workshop, I got to work with this incredible group of early career scientists: Orpheus Butler, who's a postdoc now at the University of Sydney; Seth Redman, who's now just about to start an assistant professorship at Washington State University; Celia Simmons, who's an assistant professor at UC Irvine; and Tomas Koppel, who's a postdoc at Michigan State University. And so the five of us thought that the best way to approach this quantification of interspecific variation would be to do a meta-analysis of common garden studies. And the reason why we want to look specifically at common garden studies is because stoichiometric traits, particularly elemental composition, say, are known to be quite plastic. And so because we're interested specifically in the genetic component, by looking at common garden studies, we were able to minimize the effect of the environment by comparing genetically distinct individuals within the same environment. And that allowed us, or the experimenters, to quantify the amount of genetic variation. And so with this, and this meta-analysis, we wouldn't necessarily say is like exhaustive of absolutely every common garden study that's related to stoichiometry that's out there. For example, we specifically excluded artificial selection studies, but we would describe this set of studies as a large reproducible group that allows us to answer the questions that we're interested in. So we collected information on both stoichiometric and life history traits. And when we did this, what we found was that the studies basically fell into one of two groups. They were either looking at intraspecific variation within a population or comparing representative genotypes among populations. And so in this study, we examined those two different types of studies separately, but today I'll just be presenting the differences among populations. So when we did this meta-analysis, we had a total of 30 studies, 17 which were those population studies, which I'll be talking about today. Within a given study, there could be multiple common gardens. So in the end, we had 67 of those, and the studies covered invertebrates, vertebrates, and plants, so a diversity of organisms. In the population studies that I'll be talking about today, possibly to the surprise of no one, most of the studies were done in invertebrates and freshwater systems. And our favorite study system, the Daphnia, was particularly well represented there. And what we also saw was that elemental composition was the most studied trait, and that's likely because it's relatively easy to study in comparison to some of the other traits that we considered. So what we were interested in was how much intraspecific genetic variation was present in these ecological stoichiometric traits relative to key life history traits that are commonly considered in microevolutionary studies, and do particularly classes of traits show more genetic variation than others. So when I talk about different traits, what we decided to do was group the traits that we were able to collect into various groups. So here on the very bottom, you see our life history traits, so morphology and somatic growth rate. Then we have 7 traits that are related to body composition. And then we also have this AAA, which is acquisition, assimilation, and allocation. So these are how organisms take up elements from their environment, how well they process them. And then finally, excretion. So that's how much of a given element that they're releasing into the environment. And so on the x-axis here, you can see the percent difference in trait values. So this is within a study, we would look at the difference between two genetically or two different populations. And so we would look at all pairwise comparisons within a study. And so on this section here, you'll see both the mean as well as the 95% confidence intervals and the sample size for each one of these traits. And so basically the idea is that the more variation, the higher the percentage here, the more variation there is in that trait. So when we look at these traits related to stoichiometry, what we see is that we have a fair amount of genetic variation in these stoichiometric traits. Then when we go and compare them to our life history traits, we see, however, that for many of the traits, the amount of variation is small or relatively the same as these life history traits. So there is variation there. It just might not be as much as what you're seeing in, say, morphology. The traits that exhibited the most variation were these AAA and excretion traits, whereas traits related to body stoichiometry, such as carbon and nitrogen content, showed the least variation. And so in summarizing this study, what we see is there is a fair amount of variation in stoichiometric traits. And there are differences in the amount of variation depending on what trait you're looking at. Excretion and these AAA traits showed considerable amount of interspecific variation. And when it came to body stoichiometry, there was relatively less variation, but phosphorus content, and in particular, the ratio of carbon to phosphorus showed the most variation out of these body stoichiometry traits. And so based off this study, we felt that there was at least a potential for microevolution in these stoichiometric traits. And this made me confident to move on and decide to do these experimental evolution studies. And so when we came to these studies, one of the things that I was interested in is, can the ecological stoichiometry be used as a framework for microevolution? And so this study I did with Lieben Zhao, Spiros Papakoustos, and Steven de Klerk. And yeah, Lieben is a PhD who was in my group with Steven, and Spiros Papakoustos is at the University of Turku in Finland. And yeah, he's great help with the genetic work that we did in this project. Okay, and so a concept in ecological stoichiometry is one of the main concepts in ecological stoichiometry is the idea of the growth rate hypothesis. And so the growth rate hypothesis posits that there's going to be a positive relationship between the somatic growth rate of the organism and its phosphorus content. And this is because growth requires protein synthesis, which requires ribosomal RNA. And ribosomal RNA is a very phosphorus-rich molecule. So if you're looking to grow, you need to invest in these ribosomes. And for that, you need a lot of phosphorus, so that you often see this positive relationship between growth rate and body phosphorus. And so we thought it would be interesting to see if we could use this often observed pattern to predict how body stoichiometry would change with selection for growth rate. And so here, if the expected result would be that if you have an increase in somatic growth rate, that you should also see an increase in the phosphorus content. I'll note that this is a ratio of phosphorus to carbon. So this just means the higher the value, the more phosphorus you have in your body, all else being equal, like as a percentage. And so what we would predict is, yeah, increases in growth rate should increase body phosphorus content. And so that was the goal of this experiment, was to evaluate the power of the growth rate hypothesis to predict evolutionary responses for selection for fast growth. And on top of that, we were also interested in, because we like stoichiometry, the effect of resource availability on microevolutionary responses to consumers. So if there was a stoichiometric mismatch present, how would that affect this predictive capacity? So to look at this question, we used the consumer Brachionis calliciflorus and provided it with the algae Chlamydomonas ray and hardy. And so what we did was we provided it food that was both had a high amount of phosphorus and food that was limiting in phosphorus. And so this is the high phosphorus food was about a match for the requirement for the rotifers, whereas the low phosphorus food had a stoichiometric mismatch. And so what we did was we first performed did air experimental evolution. So every genotype of rotifer will be represented by a different color. And so what we did was create genetically diverse populations. So they were composed of 30 genotypes, each represent twice. And we allocated genetically identical populations into either high or low phosphorus treatment. And then every 24 hours, we would subset the population back down to its original size of 60 individuals. And by doing so, we selected for fast population growth rates, because individuals that grow quickly are the ones that are most likely to be selected during these transfers, because they're the most abundant in the population. So we did this over the course of 35 days, and we had 5 replicates for each of these treatments. And at the end of those 35 days, we ran a common garden experiment where we took these individuals from these different treatments and cultured them both in the environment in which they were selected, as well as the alternative resource environment. And so we did this for all of our replicate populations. And what we were also able to do, which was a little bit unique of this study, was take these seed genotypes that we used here that were cultured in a completely different regime during the course of this study, and then put what we would call these naive seed genotypes into the same high phosphorus and low phosphorus food conditions, so we could estimate the ancestral state of the population. So here we see these ancestral estimates for both population growth rate in this graph and body phosphorus content here. We have the high phosphorus resource environment on the left, the low phosphorus resource environment on the right. And so with the predictions of the growth rate hypothesis, we would expect that if we had selected for fast population growth rates, that we would see also an increase in the body phosphorus content here. So yeah, this is what we would predict to see. So when it comes to our high phosphorus selected populations, what we see was indeed, we had an increase in population growth rate, and this was associated with an increase in phosphorus content. So this is perfect and matches the growth rate hypothesis. In contrast, when we look at our low phosphorus selected populations, we had a really significant increase in population growth rate, but this was not associated with an increase in the phosphorus content. So these are really contrasting results in our two different selection environments. And what we took from this was, although the growth rate hypothesis can be used as a predictive framework in certain scenarios, it's dependent on the environmental context. When we go ahead and look at these selected populations in the reciprocal environment, we see that in our high phosphorus selected populations that it performs about the same as the ancestral population. So the increased growth rate in the high phosphorus population seen here does not appear to be associated with a negative cost in the low phosphorus environment compared to the ancestral. It does about the same. And as we'd expect from the growth rate hypothesis, no difference in the population growth rate. Again, no difference in the phosphorus content of these populations. When we look at our low phosphorus populations, we see that there's a trend towards a significant increase in population growth rate relative to the ancestrals, although we have a bit of variation here. And that increase in population growth rate, again, is not associated with a change in body phosphorus content. And so, yeah, sorry, that's the wrong graph that's here, but trust me on this one, they don't change. And so that the enhanced performance of the low phosphorus populations is in the high phosphorus environment isn't associated with a change in the body phosphorus content. And so when we're thinking about using the growth rate hypothesis as a predictive framework, what we see is that although it worked in the high phosphorus environment, it didn't, and we had this, positive increase in both body phosphorus content and somatic growth rate. That did not occur in the low phosphorus, where we did see an increase in growth rate, but not in phosphorus content. And so this project was really interesting, and there's a lot of follow-up questions like what traits are used to achieve this growth rate without having higher phosphorus content. And so this is basically the type of experimental evolution project that you would run, having genetically diverse populations. But as we all know, populations aren't always genetically diverse. And so what we were interested in then asking is, can we see a heritable response to stoichiometric mismatches in populations with outstanding genetic variation? So I did this work in collaboration with Koen Verhoeven and again with Steven de Klerk. And so populations face a lot of fluctuations in their environment, whether it be seasonal changes, changes in resource availability, diseases that enter their population, and they need some sort of way to cope with this. And so they must be able to modify phenotypes on a timeframe that's relevant to the environmental change that they're experiencing. So one option is that they can exhibit phenotypic plasticity. So this is a change in the phenotype, but not in the genetic, there's no genetic change that occurs within the population. Alternatively, we can have selection on standing genetic variation, which I discussed in the last experiment. But alternatively, we can also see that there may be beneficial genetic modifications that happen within the population, for example, point mutation or transposable elements. And a more novel idea is that we may have a type of non-genetic inheritance that occurs, which change that the parent experience or the environment that the parent experiences results in a change in phenotype that gets passed on to the offspring, despite the fact that there's no genetic change. And so this could happen, for example, like with epigenetics. And so this led us to the question, as I previously discussed, of can heritable adaptation occur in the absence of standing genetic variation, as well as the absence of recombination within an ecologically relevant time frame? And so, as I said, not all populations are genetically diverse. Some are small in size. For example, invasive species, range expansions, but also populations that primarily reproduce asexually or have low genetic diversity as a result of bottlenecks that occurred due to stochastic events. And so what we wanted to look at is if and what extent heritable adaptive responses could be shown in a population, if clones differ in those responses, and if the strength of this response increases over time. And so again, we use the same system of this rotifer consumer algae that had two different stoichiometric compositions. So we have low phosphorus food. So this is bad food that is a stoichiometric mismatch, and high phosphorus food, which is we consider good and no mismatch. And we ran the same sort of selection, except remembering that these are all composed of the same genetically identical individuals. And so what we would do is, again, transfer the subset of the population every 24 hours. We were just working with smaller populations, 10 rotifers instead of 60. And we did this for 2 clones and in the 2 food treatments. And so we call this an exposure experiment instead of a selection experiment because there is no genetic diversity to select upon. We did this over the course of 55 days. And at 2 points during this exposure experiment, we ran a common garden. And so these were, again, full factorial common gardens, which we measured the population growth rate in the same way as we did during the exposure experiment. And we did these common gardens ran for 15 days. And the results that I'll be presenting here are the mean of those last 10 days of the common garden. And so if we were to see an entirely plastic response, what we would expect is that the history of exposure should have no effect on the performance of the populations, and that we would see just higher growth in our high phosphorus food and lower growth in our low phosphorus food. In contrast, if there was some sort of heritable beneficial effect during the exposure experiment, we'd expect to see an increase in these growth rates in their home environment relative to, say, the population that wasn't cultured in that environment. And so, as I said, we ran 2 clones in this experiment. So our first clone, what we see is that there was no difference between the exposure treatments, either in our first common garden as well as our second common garden. So this is a really consistent response. And that's basically what we'd expect if consistent with a plastic response to the resource environment. In contrast, in our second clone, clone B, what we see is that populations cultured in a low phosphorus environment during the exposure phase of the experiment had higher population growth rates than populations cultured in high phosphorus environment. And we see that here in the low phosphorus food, but we don't see it in our high phosphorus food. And so, yeah, there we see the difference in the first common garden, and then we see it again in the second, although there's a bit more variation, but overall in the model, these are, we see an effect. And so when we go back thinking about our research questions, if and to what extent an adaptive heritable response has evolved, we see that we do have this response in our clone B, but not in our first clone. So there's a capacity there, but it seems to be dependent on the genotype, which is the response to our second questions, if clones differ in their response. And then does the strength of the response increase over the experiment, or change in some way. It definitely doesn't increase, and we see a little bit of a change in the second common garden, but the result is consistent throughout when you look across the model. And so the implication of this result is that it appears that there's the potential for populations to adapt to environmental conditions, even when there's an absence of standing genetic diversity within those populations. And so, as I said, could be really important for populations with low genetic diversity, which are actually quite common across the landscape. And so when we're looking at this talk as a whole, there was 3 questions that I wanted to look at. The first of which is there potential for microevolution in stoichiometric traits? And I would say that from our first study, that we saw substantial variation in at least some stoichiometric traits, the greatest of which was in acquisition, assimilation, allocation, and excretion, whereas composition, which is most commonly studied, appeared to show the least amount of variation. Then when we think about taking concepts of ecological stoichiometry and using them as a predictive framework for microevolution, we saw that the growth rate hypothesis, whether or not it worked as a predictive framework, appeared to be dependent on the environment in which organisms were selected. So this provides a hint that this could be a useful tool, but we need to develop these ideas more to see where it can be really practically used. Finally, can heritable responses in stoichiometric mismatch be observed in populations with outstanding genetic variation? Yes, in contrary to the expectation that standing genetic variation is necessary for heritable responses, we did see a response in one of our 2 genotypes, but the capacity differs. And so this is also a region of further investigation that needs to be studied, as well as the mechanisms that allow for this heritable responses to occur. And so when we think about the big picture takeaways for this talk, I think one of the most important things is that adaptation to stoichiometric mismatches are able to occur on ecological timescales. So we saw this both in the second and the third talk that I presented here today. And the work that I did during my PhD, I think really just like scratched the surface of what we can need to know and what we have to study. And so there's further investigation that can be done on the traits that are responsible for the microevolutionary responses that I observed in my experiments, as well as what the ecological consequences for adaptation may be, and whether or not those changes can have potential eco-evolutionary feedbacks within the system. Additionally, I think that these studies go to show that the experimental evolution with microorganisms is a powerful tool that would allow us to unite ecological stoichiometry and microevolution a little bit more. And a lot of those questions that I mentioned previously that are still up in the air, I think can be really well addressed with this system. And so finally, I'm going to leave you with a picture of Lake Constance, which is actually the lake, if at the very beginning of the talk, that showed this really dramatic change in phosphorus availability over time. And so what I think is a nice takeaway from the work that I've been doing is, although humans are really dramatically altering these natural systems, that it does appear that both in genetically diverse as well as less diverse populations, there is some capacity to adapt to these changes, which may allow for persistence in the face of a lot of radical environmental change that's occurring. And so with that, I'm going to thank a lot of the people who are responsible for where I am today. So I'll start off with the people at the Netherlands Institute of Ecology, specifically my PhD supervisor, Steven de Klerk, who without I wouldn't be presenting any of these things that I did today. I also like to thank the PhD students in my group, Li Bin and Wei, as well as all the technicians at the NEO that helped with all these projects, as well as my funding provider, the NVO. I'd also like to thank my Woodstoic mentors for all the help and guidance they provided throughout, which was an extremely useful but also challenging process. And I'd also like to thank several people who've been really, who've helped me out a lot during my PhD. Blake Matthews, who provided me a base in Switzerland in order to finish my PhD when my partner had a job here, as well as Celia Simmons and Kyle Hudson for all their help with the experiments as well as the manuscripts throughout my PhD. So with that, I'd be more than happy to take any questions. And thank you all for listening. And yeah, that's it.
All right. Thank you, Kim. That was fantastic and really enjoyed the talk. We can have people just turn on their cameras or if you don't have a camera, you can just pipe in with a question, or you can, if you don't really want to talk in front of everyone, feel free to just type your question in the chat and one of us can read that out. And Kim is happy to answer your questions. I'm wondering if I need to. It might be that people can't actually turn like I may have to unmute people. So if you have a question,
can you hear me?
I can hear you.
Just ask it.
Yeah.
Who's up?
Hello.
So we have two questions on
your former colleague.
I think Bob had his hand up, so let's let him go first.
Yeah,
I did.
Okay, Bas or Nori?
But it's Beth and Nori. Nori is also here.
Yeah.
Nice to see you.
That was a really super interesting talk, Kim. Nori's going to change our name. I was wondering whether I misunderstood something, because you mentioned that you did an evolutionary experiment where you had a population with, or like a community with standing variation, and there you saw an effect. And then you call that evolution, but isn't that ecology if this variation is already there? Isn't it just outgrowth of one of the variants? And evolution would be change in the genotype. So there you would see a change, that would be the second experiment. That would be an evolutionary experiment. So I'm sort of, can you say something more about the terms?
Sure. So I think that microevolution would be considered genetically determined change in the phenotypic mean of the population, which is what you would be seeing in the first experiment.
No, I don't agree. I think that's an ecological change.
Okay.
An ecological change is if you see a change in the composition of the community, but their genotypes remain the same. That's ecology. An evolutionary change would be if you would see a change in the genotype with a consequent change in the phenotype or something. But if the different phenotypes are already there in standing variation, then I think it's just a shift in the ecosystem. So it's an ecological change.
Well, I would disagree. I think that there's
Stefan also disagrees.
Yeah, there's a change in the mean of the main phenotype of the population, which is genetically encoded. You see a change in the allele frequencies within the population, which is microevolution. So that would be a microevolutionary change.
But then what's the evolutionary change if those variants were already there?
You're thinking I think you're thinking of mutation. So which is a type which is evolution, but an evolutionary change is the change in the allele frequencies within a population. So despite the fact that allele was already present in the population, the fact that it is increased in the percentage of the population would be considered a microevolutionary change. But
that's interesting.
Like we have a difference of opinion there, which I'm more than happy to discuss. Yeah. Well, not opinion.
No, it's fine.
Yeah, I think it's.
I just find it super interesting, and I really like the whole approach and the setup and the results. So it's super interesting what you did. Thanks a lot.
Okay, thanks.
Nori also likes it. Okay, cool.
Great. Jim, did you have a question?
Sure, Kim. How you doing? Good to see you.
It's nice to see you too.
I'm curious about the case where the low phosphorus population didn't respond to selection for growth rate, and it's changed its P to C ratio, and but the ones who evolved under high phosphorus did. So, and I think we're thinking about a lot of these issues now ourselves. So I'm wondering, if the explanation is somehow, one of the assumptions of the growth rate hypothesis is that ribosomes function at a certain rate that's sort of constant, and I'm wondering if that might be the place to find the answer. They just have better ribosomes or faster ribosomes when they're at under low phosphorus, and when they're high phosphorus, you can afford to make a lot of ribosomes even if they're slow. So I'm wondering if you've got RNA measurements or planned, or if someone's going to do that, or.
No, we definitely wish that we didn't measure it during the experiment. To be honest, we didn't really anticipate to see this response, and so it wasn't something that we had in the initial plan. And so I think that is definitely something that we've been talking about within the group, kind of like the efficiency of which phosphorus is used by these different genotypes. And so I'm not with the group right now, but I'm sure Steven would be definitely interested in looking at that further and investigating those traits. And yeah, that's something we really want to dig into. And if I could do a zillion-year PhD, that would be definitely something I would be interested in looking at.
Going to be worth figuring out. So, but nice work, and good luck with your new stuff.
Thank you. I appreciate that. Steven, did you want to add something to that?
Yes, I would like. Yes, I would like to. Hi, Jim. Thanks for your question. Also, yeah, but I'm a little bit surprised, Kim, that you say we were surprised by that result, because it was a little bit expected. Because the reason was that if there is a lot of phosphorus available, then you can evolve fast population growth, and you can afford to need more phosphorus. So, whereas if phosphorus is low in availability, then it is a little bit kind of, well, intuitively you would then think that, or they wouldn't be able to evolve any faster somatic growth, or they would do, but they do it through, for instance, being more efficient through life history responses, for example, that they would have early maturation and grow smaller size, and at the population level you would still see higher population growth rates. But that's also what Kim checked, but that was not, there was no time in the talk, I guess, Kim, to explain that. We didn't really find evidence for that. But there may indeed be also evolutionary changes related to rates at which phosphorus is dealt with without them being adding up and contributing to higher somatic growth rate. Or yeah, so the question is indeed, it's not answered, but I was not really surprised to see that it would be different at low phosphorus content. And that's why we include that treatment as well.
Thanks. Okay. Thijs, do you have a question?
Hi, can you hear me?
Almost.
I will unplug my. Can you hear me now better?
Yeah.
My headphones have some issues. Nice to see you, Kim. Firstly,
Yeah.
haven't seen you in a while. I had a question about the selection pressure in the first experiment.
Mm-hmm.
So you say you have 60 individuals, and at each transfer you reduce them back to 60, right?
Yeah.
So how quickly did they grow? How much organisms did you have to remove at each transfer? Is that like 20% of the population? And was that the same between the HP and LP? Is it possible that you have stronger selection in the HP compared to LP, for instance? That you
sorry.
No, that was it. I was just hypothesizing.
Yeah, so that's a great question. So then the high phosphorus populations, we indeed saw larger increases within the populations. High phosphorus is just a better food. And so it's actually, so we would reduce both populations down to the starting frequency. So in our high phosphorus populations, they increased by about, they like almost 1.5%. So if it started at 60, it went to like 150, say, individuals. Whereas in the low phosphorus populations, we were seeing increases that were slightly less than 100%. And so in those ones, we were removing less individuals. So there was actually a stronger pressure on those high phosphorus selected populations than the low phosphorus ones.
I was just curious if you also analyzed the community composition at the end in both. I thought maybe you could see differences in that there, like.
Yes, indeed. Sorry, I tried to talk about a couple different things. So indeed, we did look at the genetic composition of the populations at the end. We used microsats for this. And so, yeah, it's still good enough to look whether or not the genotypes that we started with were there at the end. And so what we saw actually was that we had bouts of sexual reproduction that occurred during this experiment, so that we saw that in this experiment that either we had kind of like these very high performing populations that were part of this starting seed set, or that we had newly generated populations that occurred. So, and then at the end of the experiment, almost all the populations were dominated by a single genotype. So yeah, basically what you saw here was newly produced genotypes that led to become dominant within the system.
Thank you. And looking forward to your defense next month, Kim.
Me too.
Okay. Thank you.
So Kim, I think there's some questions in the chat. I don't know if you can see those.
Yeah, okay. So from Camilla, there's a question that it says, does it seem like the growth rate hypothesis is relevant to favorable environments, but that a different hypothesis is necessary to explain evolutionary response in unfavorable environments? So yeah, this is the idea that the use of the growth rate hypothesis as a predictive framework is dependent on the environmental context. And I would say that, yeah, maybe there's like a different hypothesis, as we previously discussed, related to phosphorus use efficiency, RNA, yeah, production, or these types of things that would be necessary in phosphorus limited environments. Okay, and then from Ziping, when you manipulate the phosphorus content of the algae, do you change the carbon and nitrogen content of the algae? Also a very good question. So the conditions in which we culture the algae do in fact change the yeah, it changes the algae. But so for carbon, we standardize the environmental conditions based on carbon availability. So the amount of carbon that's available in the experiments in the low and the high phosphorus conditions is always the same. With the nitrogen, it does change a bit, but nitrogen never becomes a limiting element. And so there are some changes, but they're not as dramatic as the ones that we see in phosphorus. And then from Patrick, is the next frontier testing how much the assimilation, these like AAA type traits can evolve, especially compared to C to P, or do we know anything about this already? No, I definitely think that's like a really interesting direction to go in. Looking at evolution in those traits is harder. The kind of experiments that you have to run and the way to assess those traits is a little bit more difficult than just body composition. But I think that looking at these traits that have the more genetic variation is definitely a really interesting place to go with these types of evolution studies. And those are also super interesting because they can lead to eco-evolutionary feedbacks as well. So and that is of course something that we're all interested in. So yeah, I definitely think that's next frontier.
All right, are there any more questions? I think we're getting towards the end here, but maybe another question or two, or if not, thank you, Kim, for giving this seminar, and good luck next month. And thank you for everybody for coming, and we hope to see you next time.
Great, and yeah, thanks again for this opportunity. I really enjoyed it, and yeah, subscribe to Ratio Matters. It's a good newsletter, world leading.
All right, bye everybody.
Okay.
Thank you.
Thanks, guys.